everyone, I am Wawa. Have you ever heard about mitosis? I am very sure that all science students is very familiar with this mitosis. So before we start the mitosis, I will explain what is the cell cycle. The purpose of cell cycle is to reproduce cell. Cell cycle has different stages. We call it as G1, S, G2 and M. M is mitosis. Okay. At the G1 is the stage where the cell is preparing to divide. It then moves into the S phase where the cell copies all the DNA. After DNA is copied, the cell moves into the G2 stage where it organizes and condenses the genetic materials or start to condense the genetic materials and prepare to divide. After all these stages, G1 and G2, we move into the next stage, which is mitosis. In the mitosis, this is where the cell actually partitions the two copies of the genetic cell. After the M phase completes, the cell division occurs and two cells are left or produced. So mitosis is the process of dividing the cell DNA into two identical daughter cells. So in the mitosis, we have four phases, which is prophase, metaphase, anaphase, and telophase, or you can remember as Pak Mat in Malay. During prophase, there are the events that happen. First, the chromatin in the nucleus condenses to form pair of chromosomes. Alright, as this is happening, the nuclear membrane breaks down to form a number small vesicles. We call it as spindle fiber. Number 2, the nucleus goes away or disintegrate because the chromosomes are trapped inside the nucleus and can't get moved to nowhere. So basically, the nucleus will disappear in this process. Number 3, the chromosome condense into compact structure. After the prophase and before the metaphase, we have one more process. We call it as prometaphase. Prometaphase, it begins when the nucleus breaks down. That means the chromosome is now attached at the spindle microtubules. This occurs at the kinetochore and results in the movement of chromosome. Where is kinetochore? Kinetochore located at each side of centromere. The next phase is metaphase. In the metaphase, the chromosomes align themselves along the metaphase plate of the spindle apparatus. Alright, just think M as the middle. During the anaphase, the sister chromatids are separated from one another and pulled to the opposite end of the cell by spindle fiber that attach to the kinetochore reagent. During the telophase is basically the reverse from the prophase. So, the nuclear membrane is reformed around the chromosome group at either pole of the cell. The chromosome uncoiled to form the chromatin. Alright, and the spindle fiber breaks down because no longer needed. The cytokinesis will take place and divide the cytoplasm into two cells of identical daughter cell. Alright, this is all.